My name is Haley Speed, and my project is using fluorescence assays to explore the regulation of the kynurenine pathway in Neurospora crassa. Neurospora crassa is an orange filamentous fungus that our lab uses to study general chromatin control processes. Specifically, I study the kynurenine pathway, which is a metabolic pathway that degrades tryptophan into anthranilic acid. Now, this pathway is really easy to monitor because of the fact that anthranilic acid is fluorescent. So essentially, you can see if the pathway is active based on the presence of fluorescent anthranilic acid or not. So in this culture, in this little figure over here, you can see that this culture is fluorescing, whereas this culture is minimally fluorescent, showing what it looks like when the pathway is active versus not. The reason why we're interested in this pathway in the first place is because it is affected by methylation of lysine 27 on histone 3, or H3K27 methylation, which is a more general chromatin control mechanism. The aim of my research is to identify any additional unknown genes that affect the regulation of this pathway. Theoretically, the genes that I identify could either be specifically affecting the pathway, which would be interesting in itself, or they could be affecting the H3K27 methylation, which would have greater implications for more general chromatin control. To do this, I conducted a primary and secondary screening of a pre-existing library of mutants that our lab has. So we're going to zoom in on this methods figure. Now, the primary screening consisted of me taking a plate out of the knockout library, copying it into another plate that contained liquid medium that had tryptophan in it, letting this plate grow up. Then from taking some readings from it, I would be able to determine which strains would move on to the secondary screening. For the secondary screening, I would grow up strains on solid medium, then transfer the tissue from the solid medium into a liquid medium culture containing tryptophan. Then I would take a medium sample after 24 hours and then take a fluorescence reading from that sample. Okay, for the primary screening, this is actually a graph from one of the 136 plates in the collection. On the y-axis, you can see fluorescence, and on the x-axis, you can see absorbance, which is actually a measure of how much tissue grew in the culture. So the ones that I would be interested in would be ones that grew enough, but also had atypical fluorescence. So this one, and maybe these two. From the primary screening, I identified about 100 strains that would then advance to the secondary screening. For the secondary screening, this is actually a table of all of the strains that I'm still studying. So there's 11 of them. And these are the ones that I determined to still be interested after the secondary screening. So here you can see is a table that shows that they replicably either fluoresce above or below wild type, which is equal to 1 on this figure. Some other things that I'm also doing is I confirmed all of the mutants by PCR just to make sure that the correct genes are knocked out. And I am also complementing all of the genes in my mutants right now, or all of the knocked out genes in my mutants right now. I'm doing this to determine if the phenotype is caused by the knockout or if the phenotype is caused by background activity that I just don't know about. Some future actions that I would like to conduct all revolve around either the question of why do my mutants have these phenotypes, or is this phenotype connected to H3K27 methylation at all? So just kind of trying to tie it back into the more general chromatin control process. Finally, I would like to acknowledge everybody in my lab for being wonderful and amazing and perfect, particularly Eric Selker, my mentor. And also I would like to acknowledge the Fire Fellowship for allowing me to really kickstart this project last summer. Thank you.